Hi, everyone, and welcome to today's webinar, Navigating Regulatory Strategies for Cellular and Gene Therapies. We're glad you're able to join us. Cato SMS is a global provider of clinical research solutions for small and mid-sized biopharmaceutical companies. Our services include strategic consulting, full-service clinical trial operations, biometrics, and clinical pharmacology. With more than 30 years of experience, Cato SMS assists sponsors in designing and executing studies from strategic planning to approval. Today, we will be discussing key regulatory strategies important to first-in-human trials for cellular and gene therapies. My name is Heather Mills, and I'll be facilitating today's presentation. Please note that some of us are working from a remote environment, and our internet connections might not be as reliable as they usually are in our offices. If you have any issues with audio or visual during today's webinar, a recording will be available afterwards, but please remain on the line. If there is a disruption, we will have things back up and running momentarily. Now I'd like to introduce you to our two presenters. Today we will be hearing from William Lee, Vice President of Cell and Gene Therapy Development, and Brandon Birch, Director of Scientific Writing and Regulatory Affairs. Dr. Lee has over 25 years of research and industrial experience in gene therapy, cellular immunology, and therapeutic vaccines, and 15 years of experience in regulatory affairs. Dr. Lee has experience in phase 1 and phase 2 clinical study designs, as well as experience in writing, design, and execution of preclinical studies, and the preparation of investigational new drug applications. Dr. Birch has over 8 years of experience with a primary focus on regulatory affairs, medical and scientific writing, and project management. Dr. Birch's experience includes overall regulatory strategy, FDA meeting support, program assessments, and regulatory submission, authoring across a multitude of therapeutic areas and drug classes, including cellular therapies. Both of our presenters will spend some time at the end of the webinar responding to your questions. In just a few moments, we'll begin. If you do have any questions, please type them in using our Q&A feature. We'll be addressing all questions at the end of the presentation. Now I'm going to turn it over to Will to start us off.
Well, thank you, Heather, for your kind introduction. So here is an overview of today's webinar on navigating regulatory strategies for cell and gene therapies. So for this webinar, I'll start by giving an overview of cell and gene therapy, touching on the approved cell and gene therapy drugs in the US and Europe. Then I'll touch on the landscape of cell and gene therapy development in today's environment. Then I'll set the stage for the current regulatory landscape in both US and Europe and talk about US and European regulatory authorities overseeing cell and gene therapy development. Then I'll discuss the important considerations for initiating cell and gene therapy, CAGT. And then after that, I will turn it over to Brandon Birch, who will talk about the target product profile as a tool to promote efficient cell and gene therapy development. So there's a wide range of gene therapy products. They can be divided into two broad groups: ex vivo delivery, where the cells are removed, modified genetically, and reintroduced back into the patient. And that's CAR T cells, CAR NK cells, or gene-modified stem cells like the induced pluripotent stem cells or CD34 cells. Then there's the in vivo delivery. That's where the gene therapy vector expressing a transgene is administered directly into the patient. And so examples are plasmids or mRNA in lipid nanoparticles, replication defective viral vectors like AAV or retrovirus, lentivirus, and oncolytic viral vectors. Then there's an exciting new class of gene therapy, genome editing. CRISPR and TALEN are examples, and they could be used either in the ex vivo gene modified cells or administered directly into patients via lipid nanoparticle. Now I'll talk about cell therapy that are non-gene modified. These are cells that are taken out of the human donor, manipulated, but not with genes, and then administered back into the patients. And these can be broadly grouped under differentiated cells. Examples are chondrocytes for arthritic conditions, fibroblasts and keratinocytes for dental or cosmetic indications. Then stem cells. Those are prominently, the example I can point to is mesenchymal stem cells for graft versus host disease, for instance. Then immune cells is a separate category of differentiated cells, and dendritic cells is the most prominent member of this group for oncology indications. And then finally, an exciting new category, exosomes, that are extracellular nanoparticle-sized vesicles that contains RNA and protein and can be used for therapeutic purposes. So here is an overview of cell and gene therapy products that are approved by the FDA and EMA, the European Medicines Agency. Here I lumped the gene-modified cell therapy of CAR-T as cell therapy, so that when you look at the pie chart in the middle, the distribution is about 3/4 cell therapy and 1/4 gene therapy that have been approved by FDA and EMA. So most of the cell and gene therapy products are approved by both the FDA and EMA, but there are a few examples here that are approved only by FDA or only by EMA. So I want to touch here on the landscape of cell and gene therapy development in today's environment. About 80% of cell and gene therapies address high unmet medical needs, either rare diseases or cancer. With the continued pace of development, the forecast is for about 800,000 patients will be treated by approved cell and gene therapy drugs by 2030. This means that now is a boom time for the emergence of cell and gene therapy companies, with the majority being in North America, followed by Europe and Israel, and then Asia. The type of cell and gene therapy products are both completely novel, game changers, or it could be incremental improvements over the currently approved cell and gene therapy products. Now I will turn over to discuss the regulatory authorities overseeing clinical development of cell and gene therapy in the US and EU. In the US, the main regulatory agency is OTAT, the Office of Tissues and Advanced Therapies, residing within CBER, Center for Biologics Evaluations and Research, within the FDA, the Food and Drug Administration. This oversight is both at the initiation of the first-in-human clinical trials all the way through the marketing application. At the local level, there is oversight by the IRB, the Institutional Review Board, but also an additional review for gene therapy by the IBC, the Institutional Biosafety Committee, if that institution where the clinical trial is being conducted receives NIH funds. The IBC's main purview is review of possible viral shedding to the environment at the preclinical and the clinical level. Now, going over to the right side of the table is the European Union, and the main regulatory agencies here are CAT, the Committee for Advanced Therapies, and the CHMP, for the Committee for Medicinal Products for Human Use. Both are agencies within the EMA, or the European Medicines Agency, and they review the marketing application at the final stage and scientific advice meetings during the course of drug development. However, different from the US, at the initiation of a cell and gene therapy clinical trial, the oversight is at the country-specific level with the filing of the clinical trial authorization, which is country-specific, not under the purview of the EMA. Then I want to talk about GMOs. In Europe, there is GMO regulation, GMO meaning genetic modified organisms. These are country-specific, and these are required before initiating clinical trials. Also in Europe, cell and gene therapies are entitled to a fee reduction for scientific advice meetings with the CHMP, particularly for small companies. And then there's also a classification procedure which allows confirmation that the cell and gene therapy product is considered an ATMP, an advanced therapy medicinal product. Finally, in Europe, the marketing approval for cell and gene therapy is through a centralized approach, the centralized process, not through a country-by-country process. And the review of the cell and gene therapy is by the CHMP within the EMA, which will then grant approval for all the EU member states. Now I'll touch upon the GMO regulations, the genetic modified organism regulations that are unique to Europe. These regulations cover gene therapies, either the human somatic cells, such as CAR-T, or recombinant viral vectors, such as AAV, adeno-associated viral vectors. The GMO regulations are directed to assessing the impact of gene therapy if it's released into the environment. And there are two categories of releasing into the environment: contained use or deliberate release. Contained use indicates that control measures are sufficient to prevent the possibility of spread into the environment. Deliberate release indicates that through the course of the clinical trial, the GMO may be spread into the wider environment. Like I mentioned before, the risk assessment for the contained use and deliberate release vary greatly from one country in Europe to another, as you can see from the table. So if the patient may shed GMO, such as AAV or CAR-T, into the wider environment, the classification of contained use or deliberate release will depend upon that country's risk assessment of the characterization of the gene therapy. For example, if a patient sheds a replication defective viral vector, the study may be classified as contained use, since the idea is that the GMO is biologically contained. So even though there's a huge disparity, EU has attempted to harmonize the risk assessment procedures for deliberate release by designating AAV vectors and CAR-T as, quote, specific risk assessment, which simplifies the procedure and the application forms for these member states. The AAV and CAR-T were singled out because they were approved in 2017 and 2018 and by both the FDA and EMA. And I think this is why they wanted to expedite the GMO procedures. Nevertheless, there's an administrative burden of filing the CTA and GMO authorization form. As you can see from the table, some are done sequentially and some are done simultaneously. The timelines can be extended if it's done sequentially. The CTA reviews generally takes 90 to 180 days, while GMO reviews can range from 30 to 90 days. So that indicates that the whole scheme of getting into first-in-human clinical trials could be delayed with GMO. Now, I'll turn to important considerations for IND and CTA submissions for the first-in-human clinical trials for cell and gene therapy. Here are some distinctive features of cell and gene therapy programs. One, gene therapy sponsors may have an additional opportunity to talk to regulatory agencies prior to initiating clinical trials. This is due to the innovative and complex trials of gene therapy. Two, compared to small molecule drugs, gene therapy manufacturing development may be extremely compressed from initiation to the marketing application. This would happen if the efficacy of the gene therapy or cell therapy is demonstrated to be large in a disease indication of an unmet medical need. Then scale-up manufacturing becomes the rate-limiting factor in development, and therefore it may be extremely compressed. Three, for the IND or CTA, regulatory agencies are open to alternative ways to demonstrate non-clinical safety for gene therapy. And finally, long-term follow-up of gene therapy is often required. So because of these additional uncertainties in development of cell and gene therapy, both the FDA and EMA provide additional opportunities to meet with the regulators. And I have on the table on the left, the United States FDA meetings, and on the right, European Union EMA meetings. But spanning both US and EU is the EMA FDA parallel scientific advice. And this is where sponsors of cell and gene therapy drugs in development can attempt to talk to both regulators for an attempt to get convergent scientific advice. I would like to point out that there is an FDA webinar very soon on this, which will give the latest developments on this EMA FDA parallel scientific advice. For the FDA, prior to IND submission, there are two meetings. The Interact meeting, formerly known as a pre-IND, which is an informal meeting to discuss product development. This is an extra meeting in addition to the pre-IND before the IND submission. Then there is the CATT, C-A-T-T, the CBER Advanced Technologies team, which is specific for discussing novel manufacturing technologies and new manufacturing methods. Then following IND submission, there's an opportunity for additional meetings with OTAT if the sponsor company has an RMAT designation, Regenerative Medicine Advanced Therapy. And this is somewhat equivalent to the breakthrough designation for other products. The RMAT designation is for cell and gene therapy products or CBER products that have a preliminary clinical evidence that has a promise to address unmet medical needs. Breakthrough designation has a little bit of a higher bar. It requires preliminary clinical evidence that offers substantial improvement over available therapies. So if you get that designation, then you have a chance for extra meetings with the FDA throughout development. Turning over to the right side of the table, early in development, usually prior to filing the CTA, sponsors can request an informal meeting with the CATT in the ITF or Innovation Task Force meeting, an informal meeting to discuss product development. And this is analogous to the Interact meeting in the US. Later in development, if a sponsor has a prime designation, then the sponsor is entitled to multiple meetings with the EMA. And the prime designation is analogous to the US breakthrough designation. And that designation is offered when there's a major therapeutic advantage over existing therapies based on early clinical data. So because cell and gene therapies often address unmet medical needs, they may qualify for expedited regulatory pathways, meaning guidance and review. On the left, I show the US expedited programs, on the right, the EU. So let's start with the US. Early in development, the sponsor may apply for fast track designation. And once the sponsor gets it, then they are entitled to a rolling review of their BLA for cell and gene therapy or priority review of the BLA. Let's say then they proceed on to development, and midway in development, the sponsor applies and is granted either an RMAT or breakthrough designation. And in this case, they're entitled to additional meetings with the regulators and the rolling review and priority review of the BLA that I mentioned before for fast track. Later in development, the sponsor may be granted accelerated approval or priority review of the BLA. Accelerated approval is approval based on a surrogate endpoint, and then there's a requirement for post-marketing data after approval. Priority review is based on a quicker review by the FDA of the marketing application. Now, turning over to the right side of the table, if the company gets the prime designation, as I mentioned before, then the company or sponsor is available for additional meetings and scientific advice meetings with EMA. Later on in development, and sort of analogous with the US, once the sponsor files for the marketing application for the cell and gene therapy, they may apply for accelerated assessment, which is equivalent to or analogous to the priority review for a quicker review by the regulators. So it compresses the normal review time of 210 days to about 150 days. A conditional marketing authorization assessment is similar to accelerated approval if it's for an unmet medical need, that's a requirement, and the benefit of patient is greater than the risk, then there's a requirement for a post-authorization data to supplement the initial data, which forms a conditional marketing authorization. And finally, the adaptive pathway is also based on a conditional approval, but this time it's in a somewhat restricted patient population. And then real world data is gathered after the approval to supplement the clinical data, and then the patient population can be broadened. Now I want to turn to some additional considerations for cell and gene therapy that's different from either the small molecule drugs or biologics. So to confirm toxicology in cell and gene therapy, for cell therapy in particular, you need to have tumorigenicity evaluations in the US and sensitive assays for biodistribution via sensitive qPCR assays in both US and EU. For gene therapy, there needs to be shown whether the gene therapy vector integrates into the host chromosome, whether there is or not germline transmission possibilities, and for the gene editing therapies, there need to be sensitive assays to determine the rate of off-target gene editing. And there's also product-specific considerations that must be taken into account. As I mentioned before, the environmental risk assessments, including GMO directives, are unique for gene therapies in Europe. And the environmental considerations in the US are under the purview of the IBC, but in a more limited scope, especially toward any viral vector, including oncolytic viruses. Now, I want to talk about long-term follow-up, which is typically required. So on the left, I put a list of all types of gene therapy vectors. And in the second column, the propensity to modify genome is the key assessment. And if there is no propensity to modify the genome, then there is no need for long-term follow-up in the US. While in the EU, it's annually for up to 5 years. For AAV, which is becoming much more prevalent in the cell and gene therapy usage, adeno-associated virus, there is a long-term follow-up of 5 years in the US, and in the EU, again, annually for at least 5 years. For the herpes virus, since it may undergo latency or reactivation, it's FDA would recommend or require up to 15 years long-term follow-up. And in the EU, from this point onward, they say that they want a follow-up at least annually until there's no risk. In the US, you notice that in the red box, it's up to 15 years, especially for lentivirus and gamma retrovirus, which are prevalently used for CAR T because they integrate into host chromosome. And then the genome editing tools, CRISPR and TALEN, since they're permanent changes in the host genome, the long-term follow-up is required for up to 15 years. So with all these considerations in mind, how is the development of recent cell and gene therapy products? So here I'm going to talk about some recent events in the last 2 years in the cell and gene therapy field and what the regulators have done. On this slide, you could see that there's a delay for CAR T for myeloma in May of 2020 due to manufacturing issues. And in August of 2020, there is a delay for gene-modified cell therapy for hemophilia A. And that's due to an FDA's request for data on the durability of the clinical response. And here I show that there's a stem cell therapy, and there's a delay. And this stem cell therapy is for graft versus host disease, and it happened in October 2020. And FDA requested more clinical data and more manufacturing data. But there's not all doom and gloom in the development of cell and gene therapy. So here in May 2021, FDA approved the CAR T that I earlier indicated for multiple myeloma that was delayed earlier, but was approved essentially about a year later. And here in November of 2020, there was a lifting of a clinical hold where FDA lifted the hold after meeting with the sponsor and agreeing for an additional potency assay. And here in August of 2021, I show an example of FDA lifting clinical hold for AAV, for spinal muscular atrophy, after the sponsor submitted a comprehensive non-clinical toxicology package. So I would like to conclude with the following take-home messages for cell and gene therapy development. The future of cell and gene therapy is bright, but the journey to approval may be rocky. Cell and gene therapies require a different drug development strategy as compared to with traditional small molecule drugs or biologics. EU and US regulations differ for first-in-human gene therapy clinical trials. And especially as I pointed out that GMO authorization is unique for EU. And FDA, EMA are taking steps to bring more cell and gene therapy products to the market by having more regulatory initiatives. So at this point, I want to turn it over to Brandon Birch, who will talk about the target product profile as a tool to promote efficient cell and gene therapy development.
Thanks very much, Will. Now that we've established a solid foundation for cell and gene therapies and how they're regulated, I'd like to introduce you to the target product profile, which is a strategic development tool that can be used to add efficiency to your drug development program and can help prevent regulatory surprises that can derail your development timelines. While this tool can be used for any drug development program, it can be especially useful for cell and gene therapy products. As Will mentioned, cell and gene therapies present some unique challenges from a development and regulatory perspective. Cell and gene therapies are fundamentally different from small molecules, including in their manufacture, dosage and administration, safety, mechanisms of action, and so forth. Therefore, it can be difficult trying to fit these products into a drug development and regulatory paradigm that was established primarily with small molecule drugs in mind. While regulatory authorities are doing a great job providing updated guidance and creating new infrastructure for cell and gene therapy products, challenges for sponsors developing these cutting-edge therapies remain. Despite this, certain fundamental drug development best practices still apply regardless of the product type. These include, among others, establishing and understanding your program goals as early as possible. This is popularly known as beginning with the end in mind, and doing so sets you up quite well for the next best practice, which is having a rational, efficient, and comprehensive plan for getting your product from early development to approval and beyond. This means understanding your product, the statutory requirements and current thinking of regulators in any regions where you would like to market your drug, and the commercial landscape and opportunities for your product, both now and in the future once your product is on the market. The next best practice is effectively engaging with regulatory agencies early. This means not only taking advantage of all opportunities to meet with regulators, including the additional interactions that Will mentioned are available to advanced therapy products, in addition to the standard milestone meetings, but also getting the most out of these interactions by providing clear and concise proposals and meaningful data to facilitate a discussion each and every time you meet with regulators. A final best practice that I'll highlight today is understanding and minimizing risks to your program and timelines. This means taking an objective, clear-eyed view of your program as it currently stands, noting aspects that are missing or otherwise deviate from what would be expected for a similar product at the current stage of development, and understanding the level and nature of these risks and their impacts on your program. So there are a few tools to consider when planning your drug development program that can help you fulfill one or more of these best practices. What I'm providing here is not an exhaustive list, and there can be some mixing and matching, substitutions, and so forth for specific drug development programs. But these tools are worth consideration because they each provide value to the development program in different ways. These include a drug development plan, which can be prepared for your full development program or for individual disciplines like quality, non-clinical, and clinical, or even more granularly, such as the clinical pharmacology plan. The next tool is a product development risk assessment, which takes a prospective look at the program and identifies anticipated or potential risks that could occur during development. This is helpful because it forces the program team to think about risks early on and allows time to think through scenarios before they happen, instead of having to take a more reactionary approach down the line. Next on the list is a program gap analysis, which is essentially a snapshot of your program at a given point in time that determines where you are relative to where you need to be. The gap analysis allows for proactive risk mitigation before the risks become serious roadblocks to the success of your program. The gap analysis can also inform critical go, no-go decisions. The last tool in the list, and the one I'll spend the rest of the presentation discussing, is the target product profile. The target product profile is especially powerful because it contributes to not one, but all of the best practices I've highlighted on this slide. It can be established very early in the development program and provides not only a firm foundation for development, but also acts as a powerful communication tool to use with regulators and others. One of the foundations of effectively applying a target product profile to your program, and the first of the best practices I highlighted on the previous slide, is beginning with the end in mind. When I hear this phrase, I'm always reminded of the quote from New York Yankees legend Yogi Berra, who said, "If you don't know where you're going, you might wind up someplace else." Now, Yogi always had an interesting relationship with words, and yet through it all, he somehow managed to gift us with some very insightful kernels of truth. And while we can be quite sure that he wasn't thinking about drug development when he spoke these words, they are nonetheless instructive for those of us in drug development. So, beginning with the end in mind. To understand the end with respect to drug development, it's helpful to take a step back and think about drug development overall. What I'm showing here is a highly simplified view of the drug development process. Depending upon where you are in the process, whether that is preclinical or phase 1, 2, or 3 of your clinical program, and depending upon what your business model is, for example, selling the asset after clinical proof of concept, or partnering with another pharma company later in development, or taking it all the way solo, wherever you are and whatever your circumstances, what you think of as the next goal will change. But no matter where you are in the process, the end goal for any commercial development program is always the same, and that is to wind up with a marketed product that meets the needs of patients, physicians, and payers, as well as the commercial goals of your company. Okay, so if the goal of a development program is a marketed product that fulfills the needs of patients and all the other stakeholders, then a good representation of the achievement of this goal is the approved product label. The product label, also known as prescribing information in the US or the summary of product characteristics, or SMPC in the EU, includes clinical, non-clinical, and quality information that profiles the safe and effective use of your product. As an example, I've included a standard table of contents for US prescribing information on the right-hand side of the slide. At this point, you may be thinking, okay, that's great. I've seen product labels before. I know what they contain. And I also know they tend to be written more toward the end of development, after all the core clinical studies have been run and the marketing application is being prepared. So how does this help me when my program still hasn't had its first clinical study yet? For this, I'd like to bring us back to the target product profile, or TPP for short. The target product profile is, again, a strategic planning tool that sponsors can use to guide the development program, including non-clinical and clinical development and formulation development, and is a powerful communication tool that can be used with regulators, investors, internal project teams, and others. To understand how a target product profile relates to product labeling, let's take a look at how regulators define the term target product profile. And here I should emphasize that on this slide and throughout much of this presentation, when I talk about a target product profile, I'm speaking primarily about what is typically thought of as a labeling target product profile. I will speak briefly about another type of target profile, one that is focused on formulation development and manufacturing in just a moment. But for now, I'm going to focus on the labeling type. The 2007 FDA guidance for industry on target product profiles defines the TPP as a format for a summary of a drug development program described in terms of labeling concepts. So essentially, the TPP is intended to capture all the important aspects of a product label and present them in a way that is organized and easily understandable. And the easiest way to do this is to set up the target product profile in a way that mirrors the arrangement of a formal product label in your regulatory region of interest. Again, for example, the US prescribing information for FDA submissions or the SMPC for submissions in the EU. Beyond the regulatory definition, it's important to reiterate that first and foremost, the TPP is a strategic development tool that should be utilized throughout the development process. As such, the TPP can and should be started as early as possible in development, allowing it to be used to inform decision-making from the beginning of a program to the end. By doing so, you not only set yourself up for success, but you're also better able to respond to study outcomes and other occurrences that may deviate from what might have been initially expected. Importantly, the TPP is a living document to which new study results, agreements with regulators, and formulation refinements are continually fed, so that by the end of the clinical program, your TPP should very closely resemble what you will submit as the annotated draft product label with your marketing application. So not only do you have a real-time view of your program status, but you also effectively address one of the requirements of the marketing application and ensure that all your labeling claims are adequately supported without any additional effort. A couple things to keep in mind, though, is that while a TPP is highly recommended, including by regulators, it is not itself a regulatory requirement. And if a TPP is prepared, it does not commit the sponsor or the regulators to anything. But simply by putting the time and effort into a well-developed TPP and discussing it with regulators throughout your development program, you can really maximize your chances of success. In addition to the labeling target product profile I just described, the other major category is the quality target product profile, or QTPP, which is defined in the 2009 ICH guideline on pharmaceutical development, also known as ICH Q8. In the guideline, the QTPP is described as a prospective summary of the quality characteristics of a drug product that ideally will be achieved to ensure the desired quality, taking into account safety and efficacy of the drug product. The QTPP is grounded firmly in the concept of quality by design, which is a systematic approach to development that begins with predefined objectives and emphasizes product and process understanding and process control, and is based on sound science and quality risk management. So in this way, it's another practical permutation of beginning with the end in mind and can be seen as a partner to the labeling TPP. The QTPP should consider all aspects affecting product quality characteristics. These include the usual suspects of intended clinical use, route of administration, dosage form, and delivery systems, but it also considers starting and raw materials, which can sometimes be challenging, especially for cellular products. There are the standard drug product quality attributes like identity, sterility, purity, and potency, and also product-specific attributes that may be important for defining the quality of a particular type of therapy. Also included are analytical methods, not only the assays, but also considering their validation or qualification and all the specifications and limits. The manufacturing process is another key aspect, including the process parameters defined for it. Container closure systems and product stability are considerations. And here it's important to emphasize that the QTPP and the quality program in general does not exist in a vacuum. So for this reason, you also need to consider things like pharmacokinetics, which is often less straightforward for advanced therapies than for a small molecule, as well as the efficacy and safety profiles of the drug. So things like the ability to connect your potency assay and results to clinical effectiveness outcomes. And then on the safety side, understanding the immunogenicity and toxicity characteristics of your product, not only for your active component, but for any excipients and impurities as well. So the QTPP can be quite involved, but these key product and process characteristics are going to be among the most important aspects for regulators when it comes to the clinical use of your product. As such, it is crucial to think about these topics early and understand how each of them contributes to the desired quality and risk profiles of your product. So I've introduced the QTPP very broadly, but I'd like to emphasize a few additional points to consider when developing a QTPP for a cell or gene therapy product before moving on. When the ICH guideline on pharmaceutical development was released in 2009, the drug development world looked a lot different than it does today. In the intervening years, we've seen the rise of many novel and innovative products. Manufacturing techniques are constantly evolving, and many more emerging technologies and advanced therapies are just over the horizon. As I noted previously, and as Will mentioned during his talk, cell and gene therapy products present additional challenges compared to small molecules. These include variable starting materials, complex manufacturing processes, challenges identifying appropriate potency assays, unique pharmacological and toxicological properties, and so forth. Because of this, when you're preparing the QTPP for a cell and gene therapy product, there are a few things you'll want to keep in mind. First, it's important to consider the interplay of product quality and risk management. At the very beginning of the process, you should perform a risk assessment, including for the starting materials, manufacturing processes, and controls, along with and in the context of the intended clinical use. You also need to think about any product-specific critical quality attributes to include in the QTPP. For example, with cell therapies, you'll likely include viability, purity, and functionality attributes, while with gene therapies, you might include the gene sequence, its expression, and the replication competence of the virus. You also need to consider the crosstalk between the quality program and the non-clinical and clinical programs, and how results from pharmacology and toxicology studies, for example, will feed back into formulation development and the QTPP. As always, as product and process understanding grows, the QTPP should be updated, and a reassessment of risk should be done at key points in the program. So that's a high-level introduction to the QTPP, and while there are many other QTPP-related considerations and topics we could discuss, I want to press forward and talk next about the advantages of including a TPP in your development program. I've mentioned several times already that one of the keys to maximizing the value of a target product profile, whether it's a labeling or quality TPP, is to prepare it early in development and to update it as new information is gathered. Doing so provides a number of advantages to the sponsor. By beginning with the end in mind, the TPP helps guide the development plan, including aiding the identification of important information needed to satisfy labeling claims, and informing sponsors on the studies, study design, and analyses that will provide that information. It also focuses the sponsor team, ensuring alignment on labeling and marketing goals, and facilitates communication and collaboration with regulators, payers, and other stakeholders. And these advantages are not just theoretical. In a study published in 2017, in which the authors looked specifically at products approved between 2008, the year after the FDA released their TPP guidance, and 2015, programs that included a TPP experienced a lower instance of refuse to file actions and a 1-month reduction in the time from first submission of the marketing application to approval. The reasons behind these improved outcomes will vary from program to program, but it is reasonable to assume that sponsors who use TPPs do benefit from an enhanced product and program understanding for both the sponsor and regulators, as well as the ability to more effectively communicate with regulators and systematically incorporate their advice throughout development. And despite this, TPPs were used in only around 11% of NDA and BLA applications under study during this time frame. According to the authors, the reason behind this low level of uptake may be, in the words of a separate group that they quoted, that there is no universal understanding of a TPP and its use. And that quote was from 2012, so even 4 years after the release of the TPP guidance, there was still a lack of understanding of what a TPP is and how it can be used effectively. Therefore, the authors concluded, it may simply be that educating sponsors on the TPP, what it is, how it is used, and its benefits, is all that is required to boost its utilization. While these data are now a few years old and look specifically at FDA's CDER and not CBER, and are presented without regard to trends at other global regulatory authorities, the findings suggest the existence of a fairly large gap between what regulators are recommending and what sponsors are actually doing. But they also highlight a real opportunity for sponsors to improve their chances of success with regulators by adopting a TPP as part of their standard drug development process. And speaking of regulators, as I've mentioned, one of the greatest advantages of a TPP is that it fosters structured, proactive dialogue with regulators. It ensures understanding and alignment on labeling claims and the adequacy of the program and study designs to support them. And in so doing, it minimizes the likelihood of regulatory surprises and resultant protracted timelines, some of the things that Will alluded to during his talk. Beyond this, TPPs can also be used to facilitate regulatory discussions about proposed promotional claims. And this can be critical, especially later in development when all eyes are turning ever more strongly to how the product is going to be presented to the world once it's on the market. And remember, again, it's never too early to start thinking about your TPP. Even before the IND is submitted, having a TPP to discuss with regulators can be very useful, allowing a sponsor to get early buy-in on their program and product characteristics. This might be at the pre-IND meeting or even an earlier engagement like FDA's Interact meeting, which we'll describe during his portion of the talk as an additional early stage opportunity available to sponsors of innovative products, including cell and gene therapy TPP in support of future regulatory engagements and to facilitate generation of the annotated product label. For those programs that do include a TPP, there are a few things to keep in mind to help you get the most out of this regulatory communication tool. When holding a meeting with regulators, it's recommended to include the TPP as an appendix to your briefing document and to ask relevant questions where regulatory input and agreement would be useful. Done this way, the TPP submission and any related discussion will be documented as part of the official meeting minutes. As usual, any agreements or advice on your product or program should be incorporated into your TPP after the meeting as appropriate. So I've spoken in general terms about what a TPP is for both a labeling type and quality type TPP and why having these tools actively engaged in your program can be so important. Now, I'd like to introduce what a TPP looks like so that you can begin to think more concretely about how a TPP can be applied to your own development programs. To start, it's important to note that there are a number of TPP templates available, including from regulatory authorities and elsewhere, and it's possible that your own company or institution already has one available. If you've seen TPPs from different sources before, you may have noticed that they can look quite different. That's because there is no one right way to format a TPP as long as the key elements are there and as long as the TPP is fit for purpose. So let's take a look at one example. Here I'm showing a couple elements from FDA's TPP template that I wanted to highlight. When using a TPP as part of your regulatory communication strategy, it should typically be arranged with a similar structure and flow to a product label, which can be tailored for the region in which you are seeking approval. In other words, like a US package insert for submission to FDA, like an SMPC for the EU, and so forth. At the top of the slide is a bookkeeping table for documenting TPP submissions to FDA. You'll notice that this table is arranged by milestones, including meetings and submissions, and that it includes essential information about whether a TPP was submitted and whether it was discussed as part of each engagement. The fact that this table is included in the FDA's template reinforces the importance of having a TPP as part of your drug development program. At the bottom, I've pulled just the indications and usage portion of the template to give an idea of how these look in this format. Again, a labeling TPP is arranged in a way that reflects the ordering of an official product label. So in this case, Section 1 of the U.S. prescribing information is indications and usage. So here, Section 1 of the TPP is likewise indications and usage. For each section, the target labeling claim is described in the column on the left, and relevant annotations, which I'll discuss in more detail in the next slide, are included in the column on the right. Also, for each labeling concept, the template includes important considerations for support. I won't read those now, but they are available in the template if you'd like to review them. While this is a nice format for sharing with regulators, it really only shows one side of the TPP, and that is the side specifically related to labeling claims. Next, I'm going to speak a bit about how a TPP can be fleshed out to provide maximum value as a strategic planning and communication tool. Unlike the example on the prior slide, a full TPP generally includes much more information than only the labeling concepts. For one thing, the TPP will typically define not just one set of product characteristics per labeling concept as we saw previously, but two, including optimal or best case criteria and minimally acceptable criteria. The optimal criteria should be based upon the realistic potential of your product given your current understanding of product characteristics, intended clinical use, and the anticipated commercial landscape. And while we all hope that our programs will meet these optimal criteria every time, if you've been in the field long enough, you understand that drug development doesn't always work that way. This is where the minimally acceptable criteria come in. These are the lower limits for your product attributes that retain the therapeutic and commercial viability of your product. If you find that one or more product attributes fall short of your minimally acceptable criteria, then you know it's time to consider whether the program can or should be salvaged, or if the best course is to abandon it altogether. These are the go, no-go decision points that are so critical to the drug development process, and they can work both ways. Not only when results come in that are less than expected, but also when results come in that are exceptionally good to the point that accelerated development, indication expansion, or market expansion may be an option. Therefore, including those decision points in your full TPP is advised. In addition to including optimal and minimally acceptable criteria for each labeling concept, the TPP should include supporting information and documentation, as well as comments that contextualize the content. Annotations typically include study details like the protocol number, the serial number for the regulatory submission in which they were included, and the submission date, and this should be done for all supporting studies. If studies have not yet been completed, then it's still important to include them here to understand exactly what parts of your clinical program are intended to support each claim. In addition to these annotations, comments should be added to provide additional supportive details. These might include meetings with the regulatory agency, with the meeting date, key discussion points and agreements, or other supportive information from your program, published literature, or elsewhere. Moving even further beyond a strictly label-based TPP, sponsors should plan to tailor their full TPP in a way that also includes aspects relevant to the eventual marketing of the product. What to include can vary depending upon your program and company goals, but often considers things like key product differentiators versus competitors, market opportunities, and valuations for each of your essential messages and claims, payer value propositions based on the market forces dominant in the regions where you intend to market your product, publicity and other outreach initiatives, including your publication plan for key studies and so on. However, I should note here that while these extra facets of the TPP will add value to the sponsor, because they're not directly related to regulatory concerns, the sponsor will want to strip out anything unrelated to labeling concepts when submitting their TPP to regulators. So here I'd like to take a few more moments to talk about some tips for preparing a target product profile. The first tip is a strategic one, because a TPP, by its very nature, requires a multidisciplinary approach. Sponsors will want to ensure that all relevant disciplines are involved in the generation and maintenance of the TPP from the very beginning. This includes representatives from project management, regulatory affairs, the non-clinical team, the clinical team, pharmacologists and pharmacometricians, the quality team, research and development, marketing, and so on. The second tip is more tactical and directly related to working within the TPP itself. When preparing the labeling aspects of the TPP, sponsors should consider all potential sections of the product label, identifying relevant sections based upon the characteristics of the product, stage of development, and so forth. Irrelevant sections may be deleted and additional subsections may be added. This will ensure that you are not missing vital pieces of information, but also that the TPP and the eventual product label are adequately tailored to your product. As you go through development and have already had prior discussions with regulators, you should note those discussions and any agreements along with the date and the meeting type in the comments section for each applicable labeling claim. As development progresses, the TPP will become more detailed and refined and with many more comments and annotations to support the various labeling claims. There will also likely be a lot of content that has already been reviewed by regulators and won't need additional review every time the TPP is submitted. In these cases, it's often helpful to highlight any new information appearing since the last time the TPP was submitted so that the reviewers can focus their attention on the most pressing information for the current discussion. Remember, for this and for any interaction you have with regulatory authorities, it's always a best practice to make life as easy as possible on the regulators. At this time, I'd like to summarize some key takeaways from my portion of the talk. As has been mentioned several times, cell and gene therapy products present additional challenges compared to small molecule drugs, including variable starting materials and complex manufacturing processes, difficulty identifying potency assays and the need for novel pharmacology and toxicology approaches, the potential for more complex clinical study designs, especially for cellular therapies, which might require surgery or various combinations of concomitant medications to be effective. And finally, given the novelty of certain advanced therapies, there is the potential that both sponsors and regulators may have limited experience with these innovative products. And there may be a lack of published guidance on key questions related to developing these products. This yet again underscores the need for early and effective communication with regulators. Despite these challenges, sponsors do have tools at their disposal to make the journey from concept to approval smoother and more predictable. By generating a target product profile early in development, sponsors gain insights into the product and program, including appropriate studies, study designs, analyses, and quality characteristics that will be needed to facilitate development and approval and ensure a marketed product that meets the needs of patients and all key stakeholders. As a regulatory tool, the target product profile fosters effective communication with regulators to clarify regulatory expectations and the adequacy of the non-clinical, clinical, and quality programs to support labeling and promotional claims. Taking this a step further, considering marketing goals as part of the target product profile can unlock additional value by ensuring alignment with overall company goals, identifying important go, no-go decision points, and facilitating effective communication and collaboration with sponsor teams, investors, payers, and other key stakeholders. Finally, to wrap up the key messages from Will's and my presentation today, in addition to the unique product program and regulatory challenges for cell and gene therapy products. We've highlighted that these therapies often require a different drug development strategy than small molecule drugs. We've also underscored that regulatory expectations can differ from one global regulatory authority to another. And we've shown you that a target product profile is a dynamic strategic development tool that can provide significant value to sponsors, especially when implemented early. So on behalf of Will and myself, it's been our pleasure to present these topics to you today. We hope you have enjoyed our presentation, that it was informative, and that you're already thinking of ways to apply these topics to your own programs. Thank you very much for your time and attention, and Will and I will return in just a moment to answer your questions. Thank you.